Have you ever wondered why drinking salt water causes dehydration or why drinking ultra pure water can be dangerous? Today's lab will give you the foundation to understand the answers to these questions. Welcome back to Biology 121, Cellular and Molecular Biology. This is Lab 2, Cell Membranes, Diffusion, and Osmosis. Histology is a branch of biology that studies microscopic anatomies of biological tissues. It's how we get all of those cool stained images of cells. Since we could image cells, we found that there are around 200 different types of cells in the human body. Cells all share 4 components. They share a plasma membrane, ribosomes, DNA, and the cytoplasm. So the plasma membrane is like an outer covering that protects all the interior biological components and keeps them safe and together. Ribosomes are molecules that synthesize proteins, and that's my area of research. DNA is the genetic component of the cell, or it's the genetic components of the cell. And last but not least, the cytoplasm is this jelly-like structure that contains cytosol, organelles, and all the other non-living components that helps us live. Most cells contain mechanisms that maintain their homeostasis. Those mechanisms are thermoregulation and osmoregulation. Osmoregulation is related to osmotic pressure. So when we talk about osmotic pressure, we're talking about the molarity, or aka the concentration. We're also talking about the gas constant, R, and we're talking about temperature. A semi-permeable membrane allows for solvent molecules to move from an area of high concentration to low concentration. This form of osmosis is how the cell maintains its balance in osmotic pressure. So here we have 3 examples of red blood cells. One where there is a balance in the osmotic pressure, and that's called isotonic, for iso, same, and tonic for tension. So that's a healthy red blood cell. The next example of a red blood cell is hypertonic. There's too little water in the red blood cell and it's all shriveled. And then the last example is hypotonic, meaning there's too much water in the red blood cell and it lyses, it breaks. These 3 examples of red blood cells relate directly to the lab. Goggles on, apron on, gloves on. Now we're all set for lab. In your lab kit, you should have a dialysis bag, a packet of iodine, a small beaker with a starch peanut inside, it's packing peanut, a sample of glucose, a pipetter, and a mixer. So I filled a cup 3/4 of the way full with warm water, so you can see some of the steam in the cup, so it's a little hotter than warm. I've cut the dialysis bag in half and soaked this part for about 3 minutes until it's malleable, so I can actually tie a knot with it. So I attempt to twist the end a little bit, and then you want to form a knot and pull really tight. You want it to be as tight as possible. Okay, now you have your cell membrane ready to go. With the extra dialysis tube, you can do another experiment. So we have 2 cell membranes. We can do 2 experiments. So the starch peanut will not completely dissolve, but that's okay. So after it's mostly dissolved, and there's only little bits floating at the top, you can add your glucose. Okay, make sure all the glucose is in, close it, and then we're going to stir that. We want to make sure all of our glucose is dissolved. And as you can see, the solution becomes very turbid / milky as the starch and the glucose mix. So now after it's all dissolved, we're good to go. For my secondary experiment, I'm going to add a package of Emergency. Emergency has salt, has a little bit of sugar, and has a lot of other vitamins like ascorbic acid, which is vitamin C. And we're going to test this in our second dialysis tube and see if iodine reacts with Emergency. Before we pour everything into our lab-made cell membrane, I thought let's find a way where after we pour it in, we can make sure it doesn't fall into solution. So I found some floss. I'm going to tie a knot and use that to wrap around the top of the cell membrane and just have the knot ready to go. So after I pour everything into the lab-made membrane, we will be ready to go. So I have one knot there, I have another knot there. Okay, so we have 2 knots. Another way to do this, if you have a paper clip, you might want to try clipping it to the side of the cup. So I'm about to pour in the starch solution, so just be careful not to pour any on the sides. I'm going to try and use this technique where you have something on the outer edge and it'll pour along the edge. So as you can see, I'm pouring very carefully. It's slowly pouring along the straw. Okay, that should be good enough. And now, while that's in, I'm going to slip my knot over the top of our cell membrane and tie a knot. Cool. So we tied it. I'm going to just double knot it. Okay. So we did the same thing with our test solution. And now we're going to tie this into a knot as well. Before you add your cell membrane to the solution, you should empty the contents of your iodine solution into the water. So now, after you've added your cell membrane into your cell environment, you'll wash it for about 15 minutes. Remember, what color does iodine turn in the presence of starch? You should be seeing a blue color, so we're watching it to see if anything turns blue. So as we watch the iodine starch solution, we want to ask ourselves, which contents are moving through the cell membrane? The options that we have are starch, glucose, water, and iodine. A semi-permeable membrane can transfer smaller molecules. Outside of the cell membrane, there's water and iodine, starch indicator. Inside the cell membrane, there's water, starch, and glucose. Water is on both inside and outside of the cell membrane, but the concentrations are different. For osmosis, water moves from high concentration to low concentration. After you've taken your bags out, you want to test the glucose level of the different solutions. So we first will test the cell environment to see if there's glucose in the cell environment. So with our indicator strips, if they remain orange, that means nothing happened. But if they turn a certain color, that means they detected what they were meant to detect. So in this case, we're looking for glucose, and we see there's glucose in the cup solution. Was glucose there to begin with? Think about it. We added water and starch indicator. So there should not have been any glucose in the solution originally. And now we also see that for our cell contents that had starch, peanut inside, and water, and now we see there's a blue color. So what reacted to give us this blue color? In our test solution, we don't see any reaction. For the final segment of this lab, we will look at osmosis in a red onion cell. Photograph A is the cell at 400 magnification before adding salt. In photograph B, we add salt and look at the same part of the onion at 400 times magnification. Record your observations in your lab handout. So after the experiment's done, you can remove your goggles. Quick note to point out with your goggles, if your string falls out, you can thread it through here and tighten it by pulling here. Remove your apron. Remember to keep your apron, it's the only one we have, and remove your gloves.